Dr. Murthy, good evening.
Good evening. How things are in Nagpur, COVID?
Yeah, about 7,000 patients per day.
Oh,
7,000 new patients.
entire Maharashtra or Nagpur?
Nagpur.
My God.
Entire
Even
Maharashtra is 50,000.
Here also things are very bad.
Nagpur is how much? 7,000?
Yes, sir,
Good
in Nagpur,
evening, sir.
7,000.
Good evening,
It
sir.
is more than Bombay.
UK is opening up, I think probably by mid-May, I think UK will be completely open.
That's what, well, I'm not sure about completely, but it will certainly become easier to pretend to live a normal life, or at least
Good.
begin to start to live a normal life. I'm not sure we'll be traveling very much or anything.
Hi, Angela. It's Hadi here. Hi.
Hello, Hadi.
Hi.
It's nice of you to spend your Saturday afternoon.
No, it's been a regular feature of Saturday afternoons attending this meeting, actually, because we
Yes,
had Kailash talking about Marsden a couple of last month, wasn't it?
I heard that. It was, I did it through YouTube, and I thought it was very, very nice.
Yeah, that's good. All due to Professor Meshram.
Mm.
Adi, nice to see you. You've had the haircut.
I have, yes.
Obviously. The last time I had one as well, but yours was needed to be done. Let's put it that way.
You know, I nearly made it into a man bun, but didn't quite get there for a pony.
Nothing escapes your attention, Rat.
Marianne, how are you? Lovely to see you.
Yeah, likewise.
Yeah. Stephen will need one. I don't think there are barbers in Allentown. Allentown is a problem.
Well, I did get one, but it's not so obvious. Sorry about that.
Oh.
Okay, Mansi, can we start?
Yes, sir. We could.
Yeah, okay.
It is 4 past the time. 4 minutes past.
Dear friends, good morning, good afternoon, good evening. Buenos dias, Banju. Tropical and Geographical Neurology Specialty Group of World Federation of Neurology and Indian Academy of Neurology have come together to organize this important series, Inspiring People in Neurosciences. In this series, we will learn about legends who have done great work in the field of neurosciences. The idea is to stimulate younger generation to do outstanding work in the field of their choice. Today, we have a 4th session, and I welcome you all for this session. Inspirational person today is Professor John Newman Davis. When you talk about myasthenia gravis or neuromuscular diseases, the first name comes to mind is of Professor John Newman Davis. He has inspired many, including the WFN president. Down the memory lane, it takes me back to 2004, when we had organized annual conference of Indian Academy of Neurology in Nagpur. And John Newman Davis was the faculty, and he stayed with us for 4 days. In one word to describe him, he was a gem of a person. Incidentally, Professor Louis Kaplan, who spoke about Professor Miller Fisher in the 2nd session, and Professor Kailash Bhatia, who talked about Professor David Marsden in the previous session, were also the faculty in that conference. To talk about this inspirational person, John Newman Davis, today we have Professor Angela Vincent, and supported by Professor David Beason and Professor Kevin Talbot. Warm welcome to you all. To chair this session, we have neuromuscular disease specialist, Professor Marianne de Visser, who is Emeritus Professor of Neuromuscular Diseases at the University of Amsterdam, the Netherlands. She graduated from University of Amsterdam, the Netherlands. She did her neurology residency at the Academic Hospital of the University of Amsterdam. And later, she did fellowship from Dr. Andrew Engel's lab at Mayo Clinic, Rochester. She enjoys working for the organization, and she is a board member, treasurer, and secretary general of European Academy of Neurology from 2014 to 2020. She is elected trustee of the World Federation of Neurology from 2002 to 2008, and currently, since July 20, she is a co-opted trustee of WFN. She was chair of the WFN nomination committee from 14 to 18. She is honorary member of Netherlands Neurological Society and Neurology Society of France. Welcome, Professor Visser. I would also like to welcome certain distinguished personalities who are present in the room. Professor Raad Shakir, former president of WFN. Professor Wolfgang Griswold, Secretary General of WFN. Professor B.S. Singhal, Director of Neurosciences, Bombay Hospital. IN President, Professor J.M. K. Murthy. WFN Trustee, Professor Steve Lewis. Professor Sarosh Katrak, Professor Hadi Manji. I welcome you all. This series is going on extremely well, and there is so much of positive feedback from the participants. I'm sure you will enjoy today's session as well. As the stalwarts will be talking about the stalwart, there will be heart-to-heart talk, brain-to-brain talk, and mind-to-mind talk. Before I hand over to Professor Visser, I would like to remind you that in the next session on 5th of June, Professor Alan Roper will talk about Professor Raymond Adams. Professor Martin Samuels will chair the session, and daughter of Raymond Adams, Professor Nina Salam, and others will join the session. So block your date. Currently, we in India are fighting with COVID-19, and we are losing the dear and near ones. Hope the situation improves soon. Over to you, Professor Visser.
Thank you, Dr. Amersham. It's an honor and a privilege to introduce Professor Angela Vincent. And this will be her bio. She is Emeritus Professor of Neuroimmunology at the University of Oxford in the UK and Honorary Professor at the University College London. She has an MBBS in 1966 and 1969. She defended her thesis, and she has an honorary position in Bergen, Norway. She's fellow of the Academy of Medical Sciences. She was awarded the Medal of the Association of British Neurologists in 2009. She became fellow of the Royal Society in 2011. She was awarded the Medal for Scientific Contributions to Neurology by the World Federation of Neurology in Kyoto 3 years ago. She received the International Prize for Translational Neuroscience of the Gertrude Reemtsma Foundation. And she shared that with Drs. Posner and Dalmau, and that was awarded in Cologne in 2018. She received the American Epilepsy Society Clinical Science Research Award, also together with Dr. Joseph Dalmau in Washington 2 years ago. She was awarded the inaugural Distinguished Alumni Award at Imperial College in London last year. And she was a vice president of the International Society for Neuroimmunology from 1996 to 2001 and president up to 2004. She was an associate editor to Brain from 2004 and 2013, and her research focuses on clinical, diagnostic, and experimental studies on antibody-mediated peripheral and central nervous system disorders. And in particular, the role of neuronal-specific maternal antibodies in neurodevelopmental disorders. Professor Vincent, Angela, the floor is yours.
Thank you. Right, thank you very much. Can I share my screen? Can you stop sharing that screen? Thank you. I can get rid of that. So thank you very much for the introduction, Marianne, and thank you, Dr. Meshram, for this invitation. It's always a pleasure to talk about John Newsom Davis, who I knew very well indeed and worked with for, I think, about 25 years. And it's been a pleasure, really, to put together some slightly different slides for this particular talk. So John was an inspiration to doctors, to patients, and to researchers. And I want to talk about his early days because I think that's quite intriguing. Changing course, laboratory research, that's the majority of what I'll talk about, and his legacies. So John actually had a not very spectacular or very happy time at British public school. That's a private school, as I expect you know. He loved literature and music and was offered a place at Cambridge to read English.
Angela, I don't see.
We are not seeing the slides.
Yeah.
Aren't you?
Slides.
Yeah.
No.
Oh, I'm so sorry. Did I not share? Share screen again. Yes, that must be the problem.
Yeah, here it is.
Okay. So as I was saying, sorry about that, my fault. I'll just
Please go on the slide mode.
Yeah, okay.
Yeah,
I know. So as I was saying, he had to defer his place at Cambridge because in those days we all did national service, or at least the men did, for 2 years, and he joined the RAF. And you can see him there very much in the middle, which was a clever position to be in. And he did that for 2 years, and that actually changed his career. So what you have to do in life is to find your métier, or in his case, it was meteor, because during that period, because of the Korean War, he was offered a chance to actually fly, which was unusual for people doing national service. And he obtained his wings, qualified as a pilot, in 1952, age 20. And he was very proud of the fact that he'd piloted a Meteor jet as part of his training. And during that period, he decided that he wanted to do medicine, not humanities. Cambridge very kindly let him change course, but this meant first attending a year in London to do science because he'd never really concentrated on any science before. And I think this was all a very important part of his development because undoubtedly the RAF had increased his confidence in his abilities. And it also showed that he could get on with people from a variety of backgrounds, not just people who'd been to a British public school. And both of those things, I think, were immensely important in his future. So from Cambridge, he went to the Middlesex Hospital in London for his clinical studies. And there he must have done very well because he was appointed to the professorial house job, which was always very much in demand. And I think throughout this period, he talked to many of his seniors about his career and eventually decided to train in neurology, largely under the influence of Michael Kramer and Roger Gilliatt. Campbell was involved in respiratory disease and the control of respiration, and he encouraged John to do some research before he continued on his training. And John was recommended to Professor Tom Sears, who was a clinical neurophysiologist doing research and clinical work at Queen Square, the well-known neurology center. And he immediately appreciated that John was earnest and enthusiastic. He had flown meteors during his national service. Tom had also been in the RAF for his national service. And together they worked. John did his MD with Tom and continued to work on intercostal muscle and the control of respiration. And there he is at Queen Square during his training in about 1964. He became clinical lecturer. He won the prize for the best MD of that year, and he seemed to be doing extremely well and continuing on in his interest in respiratory control. He also married Rosemary in 1969, and they had 3 children over the next decade, and all the children are doing very well. I know them quite well, and they had an extremely happy and fruitful marriage. So by 1970, John was a consultant neurologist, and he was directing the Batten unit, which is the intensive care unit at Queen Square, which is now called the Institute of Neurology. And he was still working on muscle spindles in intercostal muscle biopsies from patients undergoing chest surgery, usually for tumors. And by this time, he'd actually co-authored a book on respiratory muscles with Moore and Campbell and Emilio Agostini. And that was a pretty prestigious thing to have done as a young, at the time, barely qualified neurologist. However, meanwhile, just 1 mile away at University College London, there was a very prestigious department, the Department of Biophysics, with Professor Sir Bernard Katz, who was a Nobel laureate in 1970, and his long-term colleague, Professor Ricardo Mulligan. And you can see them there both. And in the middle, you can see the miniature end plate potentials, which Bernard Katz had discovered and worked on with Mulligan and with others over the last decade or so. Ricardo, however, was beginning to do something else. He was using radioactive bungarotoxin to identify and purify ACHR's acetylcholine receptors from muscle and torpedo electric organ. And he'd published 2 papers in Nature. And then in 1973, by which time I was working with him, Mulligan wanted to look at myasthenia gravis muscle, and he asked his friend, Tom Sears, whether he knew anyone who could help. And of course, John was not only working on muscle spindles himself in intercostal control muscle, chest muscle, but he was also looking after patients with myasthenia gravis before and after thymectomy in the intensive care unit. So he was wonderfully placed to take on some myasthenia research. Now, I just want to remind people of what it was like in those days, what one knew. One knew that the neuromuscular junction was an important synapse. We knew that it released acetylcholine from the nerve terminals. We didn't actually know until a little bit later that the acetylcholine receptor was a protein, but most people thought it would be. And you can see here, I hope, that the acetylcholine receptors we now know, and actually shortly afterwards we knew, were very highly concentrated on the tops of these clefts. As far as myasthenia gravis was concerned, it had been known for many years that there was a neuromuscular junction problem in those patients, and reduced miniature end plate potentials had been shown, but it was not clear whether the condition was presynaptic or postsynaptic, because nobody had been able to look precisely at the amount of acetylcholine released or the amount of acetylcholine receptors. But bungarotoxin gave you a way of doing that. And that's really what Mulligan wanted to do. And with John, we began to work on these muscles from patients undergoing thymectomy, who very kindly donated a little piece of intercostal muscle that we could work on. And over the next few years, with John and Ricardo Mulligan and Yushi Ito, we did a number of studies, and put together a pretty good paper for Brain. Unfortunately, we took rather too long over that, because by the time that had happened, we got that published, many other publications had come out on myasthenia. So Lambert et al. Had shown that patients with myasthenia have less acetylcholine receptors at their neuromuscular junctions, which we confirmed. And then Patrick and Lindstrom had showed that rabbits immunized against AChR developed myasthenia. And Lindstrom had gone on to show patients have antibodies to acetylcholine receptors. And Klaus Teucher and Dan Drachman had shown that injection of patient IgG into mice leads to acetylcholine receptor loss and small miniature end plate potentials. So all of that added up to a very nice story without us having really contributed anything very much. Those were all presented at the 5th Quinquennial Myasthenia Conference in New York in 1975. And I was very lucky that Milady asked me to go and review the conference, which was published in Nature News and Views. And I was privileged because I met all these people for the first time and was able to be known to the myasthenia gravis research community. But John had another idea, and with Tony Pinching and Keith Peters, they plasma exchanged the patient. So we knew that you can inject the antibodies into mice and the mice get weak. But what happens if you take the antibodies out of the patients? Nobody else, apparently at that time, had thought of doing this. And so John and Tony Pinching established the first plasma exchange for a patient. And this is actually the first patient you can see there. And the patient's arms could then become outstretched for much longer, just in a very simple test. And really the strength improved considerably over the next few days, and then began to get worse again after about 3 months. Now, it was really at that point that John took the plunge and applied to the Medical Research Council for a grant to start his own myasthenia gravis research, and he recruited me to join him. The lab opened in 1977 at the Royal Free Hospital, which was a department led by Professor Peter Thomas, who was world famous for his work on peripheral nerve disease. And just for fun, I wanted to add that initially it was the large wooden coffee table that was taken over for the antibody work. And the antibody assays were set up and the radioactivity was counted individually for each patient's sample on a bench monitor. So we didn't have either bench space or any equipment of our own to do the work. Nevertheless, it seemed to be successful because we started measuring antibodies to the acetylcholine receptor in patients. And you can see there the results. This was the assay that John Lindstrom had set up. It works very well. It's still used by any labs that can use radioactivity all around the world. And when we looked at the patient whose John had plasma exchange, we were able to show that there was really a wonderful inverse correlation between the patient's antibody levels and the patient's clinical strength, as you can see there. And it was only as the antibody levels came up again spontaneously that the patient became weaker again. And from there on, everything really began to get very exciting. And this was a study we did actually with Glenys Scadding and Howard Thomas at the Royal Free Hospital. There's a nice picture of Glenys on the left. She was a very energetic and bright young woman who actually became an allergy specialist subsequently. And she was actually culturing thymic cells from the thymus of patients with myasthenia gravis and wanting to know whether those thymic cells actually made the acetylcholine receptor antibody. And we were able to show that over the course of up to 140 hours in culture, the cells actually produced acetylcholine receptor antibodies and continued to do so spontaneously. So there's clearly evidence that there were B cells and plasma cells in the thymus tissue itself, which were making the antibody. I must just move, sorry, I can't see my slides because I need to minimize that, yeah. And another thing that John arranged was for us to do a collaboration with Alistair Compston. And Alistair Compston was working with John Batchelor down in East Grinstead at the time. And they were looking at HLA associations with autoimmune diseases, including multiple sclerosis, of course. And they were prepared to look at John's patients, which was great. And that study, which you see very briefly summarized here at the bottom, showed that there were really 3 types of myasthenia gravis patients: those who have thymoma, who tended to be middle-aged, and early onset, as we now call them, myasthenia gravis, and later onset patients who appeared to be different, not only in their distribution of ages, but also in their HLA associations and in their thymic pathology. And that has really stood the tests of time, and in fact, for many years was probably our best cited paper. So in 1981, we were able to recruit a PhD student, and this was Paul Whiting, who was probably the best PhD student anybody ever had. He cloned lots of antibodies from, not from the patients themselves, but from mice that were immunized with human acetylcholine receptor. And I won't go into the details of this, only to say that Paul has been extremely successful, mostly in pharma, and is now working at University College London. Meanwhile, I just did boring things like measure antibodies, and it wasn't until 1985 that we that we actually published on the diagnostic test that we were running at that time. I want to point this out because I think it was quite useful. So this is the acetylcholine receptor titers in the patients. And you see that the cutoff is here. So these are all positive, but there's a whole group of patients who were completely negative. And these clearly stood out as a separate group from the positives. And this was the first inkling that there might be a completely different antibody-mediated disease as part of the myasthenia gravis spectrum. And so with Stuart Mossman, who's now a retired neurologist in New Zealand, we were able to do a passive transfer. We injected patient antibodies from those patients who didn't have the acetylcholine receptor antibodies into mice, and they caused a defect in neuromuscular transmission, although we didn't know what was the antibody target. And it was only, in fact, much later that we were able to identify MuSK antibodies in the majority of the patients that we had tested at that time. And there's one of our key patients. You can see the atrophy in her tongue, even though she had had the disease treated for many years. Another condition, which I'm sure you've all heard about, was something that John clearly identified in the clinic. So he found that the patients with this quite well described Lambert-Eaton myasthenic syndrome had, as we knew, muscle weakness. They had autonomic signs that were important, and they improved after repetitive stimulation or sustained contraction. And of course, this condition was well known to be associated with small cell lung cancer. Therefore, it was a paraneoplastic disorder. But he recognized that some of the patients clearly had autoimmune disease and perhaps not even a cancer. And he began to think, could this be antibody mediated? And there was one good way of looking at that, and that was to give the patients plasma exchange and see if they got better. And so this is the experiment that was done. Well, first of all, you can see here in this treatment response. So here is pyridostigmine that had very little effect on the compound muscle action potential, which is always very, very small in the Lambert-Eaton syndrome. Plasma exchange, though, did seem to have a little bit of improvement. Then he stopped treating, and then some months later, he put them on patients onto prednisolone and azathioprine, which by that time he was using in myasthenia gravis. And you see over the course of a year, the patient's strength of CMAPs and strength improved remarkably. So that was one of the first cases treated like this. Meanwhile, we had a new recruit, Dr. Beason Lang, who was a biochemist, and she very heroically injected LEMS IgG into mice for up to 71 days. And we showed with Dennis Ray, who was an electrophysiologist in the pharmacology department, that the mice had reduced quantal content. So in this case, the defect was presynaptic, and it was causing a change in the acetylcholine release. That was an exciting finding. And Beth and Andy Roberts, who was doing a PhD with Beth and John, was able to show, using cancer cells from small cell lung cancers that had been grown in the laboratory, that they expressed on their surface functional calcium channels. And this is the calcium flux assay that we used. These are the results if you incubated those cells in medium or control IgG. But if you put the Lambert-Eaton myasthenic syndrome IgG, the calcium flux was significantly reduced. So we showed that small cell lung cancers express voltage-gated calcium channels and that they were reduced after incubation in Lambert-Eaton myasthenic syndrome IgG, suggesting that the antibodies should be against the voltage-gated calcium channels. And eventually, some years later, Beth and Masamoto Miura, visitor from Japan, was able to show using a cone snail toxin and VGCCs extracted from rabbit brain tissue, that there were antibodies to voltage-gated calcium channels in the great majority of patients with the Lambert-Eaton myasthenic syndrome, whether they had small cell lung cancers or not, or undetermined. And that's still a very nice assay, which is used today and is usually a radioactive assay. Meanwhile, we had also recruited Dr. Nick Wilcox as an immunologist, and it was quite clear that we were going to need an immunologist. And you must excuse me for saying we the whole time, but as you'll hear in a moment, John was so good at making everybody feel equal that there was never really any suggestion that we should say he rather than we. So Nick did a number of studies over the next few years, particularly looking at the thymus and what it might be doing. And there's a few that are summarized here. He also was very interested in the possibility of a viral initiation of the disease. So they looked in the thymus for viruses and were completely unable to find them. He then began to look at myasthenic thymus and thymoma to see if they could find AChR reactive T cells, because we knew that the B cells in myasthenia were making the antibodies, but they needed T cell help, specific T cell help, in order to make the antibody very highly specific for the receptor. And also with Miriam Schloop, who's a neurologist in Switzerland, they looked at the human thymic myeloid cells in the tissue from patients and controls and found that those myeloid cells, muscle-like cells, expressed acetylcholine receptors on their surface. It was also interesting to see what myasthenia gravis was like in other countries. And the first opportunity we had was when Jin Chu visited from Taiwan. And there were a couple of papers that came out of that, which showed that they have more young forms of myasthenia, childhood forms, and particularly ocular forms are very common in Taiwan, as in fact they are in mainland China, as we now know. But there was also some genetic differences which Nick was particularly interested in exploring. Now, I'm just going to mention something which really takes us back to the work that was started at University College London with Milady, but which then moved with John to the Royal Free. And that was the patients who didn't have an autoimmune form of myasthenia. So we did actually publish one series of 5 patients. They were all weak from early childhood. They didn't have acetylcholine receptor antibodies. They didn't respond to plasma exchange. And in this case, there were 2 examples I've shown you. Case 1 had what looked like completely normal acetylcholine receptors, that's bungarotoxin binding sites on the surface of the muscle, whereas the other case had very reduced acetylcholine receptors and very elongated neuromuscular junctions, as you can see on the left from the cholinesterase staining. So this suggested that there was going to be heterogeneity of some syndromes, which almost certainly genetically caused. And it was for that reason that we were very lucky to recruit a molecular biologist, Dr. David Beason, who you're going to hear from later, who will tell you all about the cloning of the human AChR genes and the genetics of the congenital myasthenic syndrome. We also wanted, we were really quite demanding, we wanted David to make recombinant proteins of the acetylcholine receptors for the T cell studies. And that he did extremely willingly and very productively and in parallel with his genetic studies. So everything had been going swimmingly. In 1979, John had become the first MRC clinical research professor. The MRC supported us with 4 program grants. We could have stayed at the Royal Free. But in 1988, Professor Sir David Weatherall, world-famous geneticist and clinician, invited John to apply for the Action Research Chair of Clinical Neurology and to establish research in the new Institute of Molecular Medicine, now called the Weatherall Institute of Molecular Medicine. And by that stage, there were 4 of us, myself, Nick, David, and Beth. Martin Bridson had been recruited and in fact helped with the transfer of all our sera and cells and blood cells and thymuses to Oxford. And Kerry Mills was a clinical neurophysiologist. Laurie Lowe, who was the anaesthetist doing the thymectomy operations, and Eve Goodyear, who was a great myasthenia gravis nurse and plasma exchange expert, those 3 also all came to Oxford with us, plus about 12 other people in the lab. So it was a sort of emigration from London to Oxford. And that was a very productive time, particularly for the T cell work that Nick was doing. Ben On, who subsequently became the head of the Director of Medical Services in Singapore, more recently, of course. He did a lovely study with Nick predominantly and some of our new colleagues in Oxford, where they showed that they could clone and characterize a highly acetylcholine receptor specific T cell from one particular female myasthenia gravis patient. And that was really the first complete characterization of a T cell specific for the acetylcholine receptor. In 1992, John was elected to the Royal Society, a fellow of the Royal Society. And we said, look, you're not getting away with this. You've got to recognize how many people have helped. And so we had a group photograph, and you can see John there in the middle with many of the people who had really helped. But also, this was a very exciting time, not only for John and his research, but also because of the number of foreign visitors we were beginning to acquire and who were very instrumental in helping with all sorts of research projects. So one particular one was Simon Hawke, who's now a clinical neurologist in Sydney. And he showed that you can use very highly purified acetylcholine receptor and that it was a very good antigen for T cells and was able to stimulate specific T cells at much, much lower doses, lower concentrations than any of the recombinant proteins or peptides that other people were using. And I think this was an important advance, which unfortunately many people didn't really bother to follow on from, which was sad because actually they were all using peptides and were getting some rather irrelevant results, as Nick showed. And then there was Mike Nichols, and he was from Canada. He had a Canadian MRC fellowship to come and work with us. And he showed that you can kill AChR-specific T cells by a MHC peptide complex, if it's the right peptide. And that was a peptide that we had identified was stimulating the cells that actually recognize the acetylcholine receptor. What happened was that the specific T cells would become apoptotic, and this was potentially a very nice potential treatment. And that was really what we were trying to do, is find a way of treating myasthenia in a much more specific way. However, it was pretty hard work, and one way and another, it didn't in the end get to the clinic. And that was rather sad at the time. We needed to know how many patients had any of these specific identified T cells. And there was one hope for finding, which Margaret Hill was able to show in her PhD work. She discovered a new T cell epitope, which was common to patients with early onset myasthenia and restricted to the epsilon subunit of the mature acetylcholine receptor. And this looked like something that was really going to be important. But again, it was difficult to follow up to find more patients with the same T cell. And in the end, we were not able to take it much further. But we were able to clone some antibodies, and that was actually led by Jeremy Farah, who, as probably many of you know, was previously director of the Oxford University Clinical Research Institute in Vietnam, and is now director of the Wellcome Trust in the UK, and a great expert of infections. But at the time, he worked with us on myasthenia, and he was able to clone and characterize human acetylcholine receptor antibodies using the techniques that were available at the time. And I won't go into the details, only to say that not all antibodies are the same, as we had suspected from previous work, but he showed that very nicely. But something which goes back to the T cell side was a study done by Camilla Buckley, who's now a consultant neurologist, an MG specialist in Oxford. And she was able to show that thymomas generated long-lived T cells that were unusual in normal patients, and that those T cells rose during malignant recurrence and were associated with antibodies to cytokines. And that's antibodies to cytokines is something that Nick Wilcox took forward and continues to be interested in to this day. And this was a nice paper published in Annals of Neurology, but in terms of clinical usefulness, it wasn't something that was taken up widely. John still had at the back of his mind that he ought to identify some more autoantibody-mediated disorders. And in fact, there was, of course, one which is acquired neuromyotonia. So he recognized that these patients had what might be peripheral nerve disorder, might be a genetic disorder, and probably is in some cases, or even toxic disorder, but that some of the cases had autoimmune diseases, suggesting that they might have an autoimmune form of neuromyotonia, and also some of them had tumors, particularly thymomas. And so we did a study done by Dr. Sinha, which showed that if you inject patient IgG into mice, they show resistance to curare. So they actually had improved neuromuscular transmission, which fitted nicely with the electrophysiology that Kerry Mills had shown, which is shown on the top right, of these multiple neuromyotonic discharges. And at the bottom on the right, you can see that as you plasma exchange the patient, the mean discharge rate per second fell remarkably on the 2 occasions as it happened in that particular patient. So this all suggested that the patients with neuromyotonia may have antibodies, and we began to suspect that they had potassium channel antibodies, and that's what we worked on for some time before we realized that actually they were antibodies binding to other proteins that complex with potassium channel. And a lot of that work was done by Ian Hart, who became a consultant neurologist and neuroimmunologist in Liverpool, but very sadly died not very long after John did. So this on the right is a patient with neuromyotonia, and I hope you were able to see that. So there was another aspect which takes one back to the acetylcholine receptor, which was important towards the end of John's time in Oxford, or at least his official position in Oxford, which was the association of arthrogryposis multiplex congenita with maternal antibodies inhibiting the fetal acetylcholine receptor. And that was clinical work done by John and others, but in particular, it was essential to have the help of David Beason and Claire Newland, who was an electrophysiologist, who actually showed that the IgG from these mothers inhibited the fetal form of the acetylcholine receptor, but had no effect on the adult form. And I hope you can sort of briefly see that to the left. We then were able to transfer the disease to mice using injecting the maternal IgG in serum actually into pregnant mice and showing the effects on the fetuses. And that was performed by Louise Jacobson, who is still working in Oxford. So what were John's greatest research achievements? The first use of plasma exchange in myasthenia and other diseases, the role of the thymus in myasthenia and different clinical groups with Compton, Scadding, Wilcox, cloning of specific T cells from patients and development of tolerizing approaches for human disease. Wilcox, Nichols, Hawke, Hill, all contributed enormously to that. The Lambert-Eaton myasthenic syndrome caused by antibodies against calcium channels with Beth Lang, but also with Andrew Engel in the USA, with whom we had many collaborations. The fact that acquired neuromyotonia could be caused by antibodies, which we thought initially were binding to the potassium channels with Ian Hart, Peter Molyneux, and others. And of course, defining congenital myasthenic syndromes with David Beason that you're going to hear about in a moment. So what made John so special? Well, he was clearly highly intelligent, enthusiastic, tolerant, unpretentious. And those 2 last qualities, I think, were particularly important. He inspired confidence in everyone who worked with him in the university and NHS, inspired loyalty in all who worked with him in the lab, and in the individual patients who willingly provided samples and underwent experimental treatments, which plasma exchange was in those days. And he supported others' talents. He allowed autonomy as appropriate, and he treated at least the sort of core lab team as equals. When he came into the lab, he would divest himself of his tie and jacket and change into casual clothes. And then we were all on an equal footing. And of course, over the time, he hosted many foreign visitors, and he forged friendships with doctors and patients all over the world. Post-retirement, as you're going to hear much more of from Kevin Talbot in a moment, was to continue the clinics. He was president of the Association of British Neurologists and editor of Brain for 7 years. And his legacy, of course, is in the department that was expanded to include other fields and expertise. A neurosciences team that continued to work together, retirements permitting, and in fact, many of us still do work together, even despite retirements. And neuroimmunology research in particular that continued until 2016 and is now led by Drs. Sarosh Irani and Patrick Waters in Oxford. So that was a picture now 16 years ago, before David Beason's group, which there's David, stayed in the Weatherall Institute while the rest of the neuroimmunologists went to the other end of the hospital, which is where they are now. But the other legacy, and I just want to quickly go through this, is the thymectomy trial. So as you can see at the bottom, if you only thymectomize patients, give them no other treatment, their antibodies and their clinic do drop and their clinical grades do get better over time, but it takes about 10 months to get a 50% reduction. If you just give them steroids on the right, you can get the same effect in about 5 months. So is thymectomy really necessary? That was the question John asked, and you can see his rather quizzing expression on the face there. And I won't go into the details of how long it took to set this trial up. It was incredible that John achieved it. Eventually, he had a wonderful team from the USA. He was the only European, actually, in the executive committee. Gary Carter, Gil Wolfe, Henry Kaminsky, Robin Conwit, and Alfred Jurecki, who actually has already died, unfortunately, but at a grand old age of 94, but was the surgeon who really made sure that thymectomy was done in the appropriate manner. Complete thymectomy is so important. They had a wonderful time. John so enjoyed it with these people. He made great friends with them all. And they had a clinical trial comparing New Zealand with Californian Sauvignon Blanc at the Oxford Myasenia Gravis Thymectomy meeting in 2004. And he was very pleased because he was able to choose the New Zealand rather than the Californian Sauvignon as the best one. And I think they were all rather cross. But you can see that they're all enjoying the occasion. During that period between 1998 and 2007, and in fact, particularly between 2002 and 2007, he made altogether 57 trips to 46 different destinations for lectures and another 16 or more trips just to tell people about the myasthenia gravis thymectomy trial and to help make sure that they had the right conditions for doing the thymectomies. There were a massive amount of travel that he did at that time. And sometimes he was able to enjoy meeting up with old lab people. These are 5 Japanese postdoctoral fellows that have visited our lab. They're all clinicians. They're all now doing very well back in Japan. And these were occasions which John was wonderfully good at, very good at talking to everybody, very good at sharing the situation, like kneeling on the floor, which I found very uncomfortable, I must say, but he was great. He managed to help recruit patients from all over the world, and eventually there were 126 patients from 67 centers in 18 countries from 6 continents, which is a massive achievement to recruit those patients and to get so many centers to be involved in the trial. And of course, the trial was successful. Thymectomy led to improved quantitative myasthenia gravis scores and reduced requirement for prednisone. And they were published, unfortunately, of course, posthumously for John in 2016, and then a repeat in 2019. So I just want to, if I may, just say or really quote from my own speech at his commemoration. My colleagues and I have spent many hours thinking about John and what was so special about him and how he achieved so much. Others talk about his energy, but in fact, he seldom appeared to hurry and managed to maintain a casual, almost sauntering style in all he did. We wonder at his extraordinary tolerance and ability to devote time and people at every level, making each of them feel that he was their friend and they were his equal. And his enviable eloquence meant that he could diffuse any situation, however tense. And I suspect that we subconsciously try to emulate him in our daily lives. So thank you for listening. I'm hoping now that you will be able to enjoy presentations from David Beason and then Kevin Talbot.
Thank you very much, Angela, for a beautiful overview on the life and the career of John Newsom Davis, who, as you mentioned, not only inspired his collaborators, residents, and students, but above all seemed to have been a doctor who used the knowledge he derived from his extensive and exciting research to treat patients. I'd like to introduce Professor David Beason now. Who graduated from Magdalen College, Cambridge, and specialized in genetics. He obtained his PhD at Imperial College in London, focusing on the first cloning of muscle acetylcholine receptors. As we heard from Angela, he moved with the group of John Newsom Davis and Angela Vincent to the Wetherell Institute of Molecular Medicine in 1988 to work on disorders of neuromuscular transmission. He was made a professor of neuroscience in 2004 and became fellow of the Academy of Medical Sciences in 2017. He was the first to clone many of the genes encoding key proteins at the human neuromuscular junction, and more recently, his research has focused on identifying and understanding the genes and molecular mechanisms that underlie congenital myasthenic syndromes. The research has involved close clinical collaborations, and the success of the work enables the National Commissioning Group of the NHS to make Oxford the National Centre for a Diagnostic and Advisory Service for these disorders. David, the floor is yours.
Right, just trying to share screen. Is my screen up?
No, not yet. Yeah.
Right. Okay, so thank you very much for that introduction. And I'm just going to give a very brief view of a scientist working under a clinician, a great clinician in John Newsom Davis. And he had this view, which is obviously a valid view, that patients actually seeing what happens in patients can inform us about what's happening at the neuromuscular junction. And he liked to utilize his clinical expertise and his astute observations of the patients and the patterns of the patient's weakness and their history to sort of get ideas about actually what might be happening at the neuromuscular junction. And he used to combine this with a great love of neurophysiology to actually continually think in the clinic about what's happening at the neuromuscular junction within his patients, and therefore what the potential therapies were. And also just working under him, I haven't got this up on the slide, but as Angela mentioned, he originally was going up to Cambridge to study English. And he had this magnificent facility for words and was able to, if you wrote something and you would spend sort of 2 sentences, 3 sentences trying to get your meaning across, and he'd come back with just the perfect word, getting your meaning across in one very short, concise sentence. So his ability to write English was fantastic, I mean, amazing. And it was very helpful for us scientists in the lab to have someone who could teach us how to write papers and how to get your meaning across. So that was something very useful. But can I, whoops, sorry, I'm trying to,
Down on the slide, there will be arrows. So
has it come on the slide now?
No, the
Can you
next
see
slide is not there.
The bottom left, there will be arrows. You can click anywhere on the slide.
I don't seem to have got it on my.
If you click on the slide, it sometimes works just like that, dude.
All right, has that come on?
Yeah.
Yeah.
All right, brilliant. Okay, so what he was a great believer in was, as it were, how you could go from the bedside to the bench and back. But, and usually, in fact, often in so many sort of labs or research labs, you try and train clinicians by sending them do their PhD or their other short research periods in the laboratory to work out sort of how science works and therefore how you can translate what happens in the laboratory to actual clinical practice. But John actually sort of took the, as it were, the reverse idea, or this was his sort of expanded thinking. And he thought, actually, what happens if we take a scientist and take a scientist to the clinic so that they can view what goes on in the clinic, see the patients, and then go back to the laboratory and think about what experiments to do. So this is sort of the reverse way around of what's normal, usual practice in medical science. So in fact, he took the scientist, and I was lucky enough to be that scientist, to actually attend the clinics every week to see the patients and then be able to think about what is actually going wrong in the patients, go back to the laboratory, do some research, and then come back to the clinic with thinking, what's wrong with this particular patient and how can we treat them? And typically just in that slide there, you're looking at some recordings of mutations in the acetylcholine receptor, which is we have sort of been working on for a long time to clone. And you can actually see the sort of second bottom. In fact, looking at those openings of the channels, we can see they're open for a long period of time. And therefore, we want a treatment that's a channel blocker. So in this case, one can use fluoxetine. At the very bottom there, you've got the burst durations are much too short, i.e. this is a fast channel syndrome. And therefore, in the treatment, for this case, we would use pyridostigmine or something to actually enhance neuromuscular transmission rather than to inhibit it with the fluoxetine. So essentially what he did was able to sort of put a scientist in the clinic, and then actually that provides the scientist and all the team with the enthusiasm to go back and think about how we can work out what's happening in the patient and then go back to the clinic. He also had the sort of prescience to realize that for rare neuromuscular diseases, actually having a cohort of patients that's very well clinically characterized is absolutely essential. And that if you have a cohort of patients that's extremely well characterized, it makes the life of those in the research and the research laboratories so much easier. And so he was instrumental in being part of the national service, or setting up a service for congenital myasthenic syndromes that induces all patients or provides all patients with a potential myasthenic syndrome all around the UK to be able to be referred to Oxford. And then we can study numbers in higher numbers and therefore have a cohort that provides for a decent scientific investigation. So having done that, perhaps the most important thing that's evolved over the years coming from this research was actually a group of patients who had mutations in a gene called DOX7. Now, DOX7 is a protein at the neuromuscular junction, And there's a pathway at the neuromuscular junction where agrin's released from the nerve. It complexes with a protein called MuSK, which signals to other proteins at the neuromuscular junction to actually form the neuromuscular junction and cluster all the receptors in the right place. And the DOX7 protein is a sort of amplifying signal. So the MuSK signal on its own is very weak. But if you've got DOX7, it amplifies and clusters the receptors in the right place very efficiently. But also what was being found is it's crucially important in providing for synaptic structure. So it has a key role in ensuring the structure of the synapse is correct. And so what's been discovered over the last, 10 to 15 years or so is that, in fact, it's almost as important to have the right structure at the neuromuscular junction as to just have it working well. It's got to be working well and it's got to have the right structure. And I can remember when we first identified mutations in DOX7 and I emailed John and said, I think we've got the limb girdles here. And I can remember his excitement coming back. This was at a weekend and he was, I think even on Saturday evening, he was emailing back saying, oh, let me tell me all about it. So he sort of was always there, able to be contacted and enthusiastic about finding out what was going wrong at the neuromuscular junction. Anyway, so this is crucial a role. And in fact, what's evolved from this is actually because we had the cohort of patients together, we very quickly saw that these patients responded or didn't respond to the standard treatments, which was pyridostigmine, but responded incredibly well to ephedrine. And in fact, they responded quite slowly to ephedrine, but the response is absolutely remarkable. And what we saw was that patients could get out of their wheelchairs and start walking. From being very disabled, gradually they became stronger and stronger and went from wheelchairs to walking, which was absolutely remarkable considering previously this hadn't been revealed. So what's been, I think, evident here is that actually this ephedrine actually has a role in restoring the structure of the neuromuscular junction that is defective if your DOX7 isn't working correctly. And if you can restore the structure, then you can make a remarkable effect on treating the patients. And in fact, it's not just patients with DOX7, but what we have been able to go on to show since is that with salbutamol or ephedrine, you can actually, if these patients with receptor deficiency or other forms of the congenital myasthenic syndromes are treated with pyridostigmine, which works very efficiently and effectively initially, actually over time, in many cases, the effectiveness wears off. But if you then add in the salbutamol, you get another remarkable effect for treatment. So what is apparent is that, again, the pyridostigmine is actually detrimental to the structure over time of the neuromuscular junction, but with the beta adrenergic receptor agonists, it can be restored. And even so well that sometimes you can even get in the papers for the remarkable effect that it can have on your patients, as shown here by an article in the Daily Mail. What's also become apparent is that, as Angela mentioned, the myasthenic syndromes aren't just a few proteins. There are lots and lots of things that can be affected. And they can be affected by proteins that aren't necessarily just located at the neuromuscular junction, but are found more widely in the body. In fact, some ubiquitous proteins like those involved in end-linked glycosylation. And for some reason, we're not quite sure why, proteins found in the very early stages of the end-linked glycosylation pathway can cause congenital myasthenic syndromes. Some severe mutations in these genes cause congenital disorders of glycosylation, which is very severe, but the relatively mild mutations, which are affecting perhaps the level of expression of the proteins, actually it can just cause a syndrome which just has the myasthenia symptoms and doesn't have the wider severe multi-system disorders that you can get for the congenital disorders of glycosylation. So again, it was the ability or the legacy of John to produce these cohorts of patients that's allowed us to pick these out, so that now, in fact, following on with the advent of the genomic exome sequencing and whole genome sequencing, many proteins have been shown to have an effect at the neuromuscular junction. The original proteins with mutations in them, such as the acetylcholine receptors and rapsin or MuSK, which are specific to the neuromuscular junction, were the obvious ones. But now what people are finding are lots of proteins that don't necessarily specifically localize to the neuromuscular junction, but actually have an effect there. So perhaps treating proteins that are localized, or treating patients with mutations in proteins that are localized to the neuromuscular junction, is relatively easy. But what's going to be proved much more difficult in the future is these proteins which have a multi-system or many effects. So they may have a problem at the neuromuscular junction, but if you've got something that's affecting acetylcholine release from the presynaptic terminal, then that's going to affect all the cholinergic systems, and therefore you're going to have multiple problems, say mild learning difficulties or other problems. So that's going to produce a more challenging way in which to look at how we can treat these in the future. So what for the future? Well, one is obviously gene therapy, and it's now a very exciting time for neuromuscular disorders with gene therapies such as those for SMA coming through. In the future, perhaps we'll get a cure with gene editing. And then one final thought is maybe we can use the biology of the neuromuscular junction to generate treatments. And just one thing we're interested is this phenomena with DOX7 is that if you overexpress DOX7, which is shown on the right, normal neuromuscular junctions, you can see they're labeled with the fluorescent snake toxin, bungarotoxin. And the bottom, if we put in excess amounts of DOX7, we get these hugely enlarged neuromuscular junctions that in animal models of disease actually work very efficiently and essentially act as quite a good treatment. But perhaps also this could be used not just for disorders of the neuromuscular junction specifically, but other disorders that one gets, such as sarcopenia, where perhaps if we can enhance the neuromuscular junction, we can help the patients. So we think that's sort of, it's now building on John's legacy, an exciting time at the neuromuscular junction, and sort of my work and many labs around the country have learned from what he's done and how he's helped us with the study of these disorders. So just like to say thank you to John Newsom-Davies, and just showing labs here that are continually working, at the bottom a mixture of clinicians and lab workers, and on the right lab workers who are working hard to take what John started off and take it forward. And I'll finish there. Thank you.
Thank you, David, for sharing your memories on the exciting research you carried out with John Newsom-Davies, either in person or through the patient material collected by John. Thank you very much. I'd like to introduce Professor Kevin Talbot now, who is head of the Nuffield Department of Clinical Neurosciences at the University of Oxford. He graduated from medical school in 1990, obtained his doctorate in 1998 on the thesis on molecular pathogenesis of spinal muscular atrophy. Finalized his neurological residency in 2000 and became a fellow of the Royal Society of Physicians in 2006. Kevin is a clinical neurologist specializing in neurodegenerative diseases, especially motor neuron disease, and in particular ALS. He's director, the Oxford Motor Neuron Disease Care and Research Center, professor of motor neuron biology at the University of Oxford, honorary consultant neurologist at the John Radcliffe Hospital in Oxford, and fellow at the Pembroke College in Oxford. His research group studies the molecular basis of motor neuron degeneration using mouse and stem cell models with the aim of identifying effective treatments. Evan, the floor is yours.
Thank you, Marianne. Right. Well, I thought I would actually just take the opportunity to talk a little bit about John in the context of Oxford history. Most people will tell you that any conversation in Oxford starts with a history lesson. This is the Radcliffe Infirmary, which was a hospital opened in 1770, and it closed as a hospital in about 2008. And now let me see, we're having, oh, there we go. So Oxford has a long history of neuroscience. So here is Thomas Willis, who coined the term neurology here in Cerebri Anatomy, famously described the circle of Willis, but actually really brought together clinical description with the anatomical and physiological method to really explain why disorders of the brain led to the signs and symptoms they do. So much more than just coining the term neurology. This is the 400th anniversary of Thomas Willis's birth. And if you're interested in reading more about this, there is a website in Oxford. You can look at this with various resources, with interviews and exhibitions. Alistair Compston later on this year will publish the biography of Thomas Willis. An amazing figure, really. Another major figure in Oxford neuroscience, Charles Sherrington, Nobel Prize for Physiology and Medicine, really, who developed our view of the synapse and the function of the nervous system at the very fundamental level. And I mention this because actually it was only, despite this sort of background, it was only in 1966 that the first chair in neurology was developed in Oxford, and that was Ritchie Russell, and this is the Radcliffe Infirmary probably at the beginning of the 20th century. And he, interestingly, had developed his research on head injury during the Second World War, and also had a great interest in polio. But really, it wasn't really until then that neurology was recognized in Oxford medicine as a sort of academic speciality. Brian Matthews came after Ritchie Russell, and he was a very influential figure in clinical neurology and was thought really to be the best clinical neurologist in the country at his time, but also developed epidemiological research into CJD and developed Oxford as a stroke research centre. So that is the sort of context in which John arrived in Oxford. And as Angela has said, he was more than a clinician, he was also a scientist. And that's really the pivotal point, I think, in the development of Oxford neurology when John arrived. And that, for me, as somebody who did a DPhil in Oxford and trained in neurology, John was an absolutely crucial figure in teaching me that you can do both. And it's important that some people do both. And on the background of this really quite good clinical department, David Weatherall wanted to bring in somebody who could develop clinical neurology as a scientific discipline. And David Weatherall, when John died, I think, said that he had fulfilled this completely. He had expanded the high standards of clinical neurology established by his predecessors, but at the same time established molecular and cellular biology in the neurological sciences. And that's very much the model that David Weatherall was trying to develop in Oxford across all specialities. And this was done, as Angela said, at the Weatherall Institute for Molecular Medicine. And it remains a powerhouse of medical research. And there are now 3 or 4 or 5 different buildings like this around Oxford, where I think people coming through who are training in any area of medicine can realize that an active clinician like Peter Radcliffe here can win the Nobel Prize if they are in the right environment where their in-depth understanding of a disease can lead to scientific advances. So John was a clinician, and he developed a clinical speciality clinic in Oxford for myasthenia gravis. And what it taught me was that if you really want to understand a disease, you really want to study it, you need to offer something to the patients who will be your partner in research. And that's very much the way that we developed the Motor Neuron Disease Center here. And he recruited, when he came, people to support this in clinical neurophysiology, Kerry Mills, as Angela's mentioned, but also developed the neuropathology department as an academic department. He brought in Peter Rothwell as clinical lecturer, who's gone on to develop a very well-known stroke research program, and importantly, Paul Matthews as head of imaging. So as Angela's pointed out, this is a paper from 2016, I think. Long after he died, he was still appearing in the literature. So in terms of clinical research, his reach extended for a long way. As well as neuroscience, Oxford was one of the places where MRI was first developed. And Oxford Instruments developed the first commercial spin-off company of any kind from the University of Oxford, and they developed the first superconducting magnets that allowed MRI to occur. And the first commercial MRI body scanner was manufactured in Oxford. And the reason that's important is that together with George Rada and Alan Cowie, John Newsom Davis had the vision to realize that if you could embed a research scanner in a hospital, you could achieve a lot in terms of understanding how the brain functions. So they developed this center called FIMRIB, the Functional Magnetic Resonance Imaging Center for the Brain. And they started producing functional MRI data. This has now turned into probably one of the most important centers for imaging research in the world. It's now called the Wellcome Center for Interpretive Neuroimaging. It's within our department, Nuffield Department of Clinical Neurosciences. And it's got, approaching 150 people who are at the forefront of developing how imaging can really teach us about the brain. I think this is a tremendous legacy of John's time. He had the vision with others to develop this very important center. There are many people who would have spoken positively about his influence on them. Jackie Pallis and Camilla Buckley are two that Angela has mentioned. And you can just quickly read through these statements, but what they tell you is that his personal qualities were immediately obvious to anybody who came close to him. I had the privilege to see him as a mentor. I went to him for advice very regularly, even though I wasn't working in the same field, clinically or scientifically. And he, as Angela has said, had that amazing quality of personal charm. He put you at his ease, but also, you could tell you were dealing with somebody who was of enormous stature, who commanded respect. And that's a combination which is very impressive. So how did he do it? Well, despite being academically very active and strong, he chaired the grand round every week. So that was an important message that the head of department was there, supervising the communication of clinical neurology for next generations through case histories and presentations. He, until retirement, continued to be on the on-call rota. So my very first Saturday evening as an on-call registrar, which was pretty terrifying, I was on call with him, and I was faced with a patient in a scanner at 8 o'clock in the evening who was basically coning with a normal scan. And the neurosurgeon said, well, it's over to you because this must be neurology because the scan's normal. And I really had no idea what was wrong with this patient. So I phoned him up and he came in and he immediately told me what was wrong with the patient. And he was right. It was somebody with ADEM. And that just made me feel I was working with somebody who, A, was a fantastic instinctive clinician, but also immensely supportive to somebody at the beginning of their neurological training. Editor of Brain, well, again, you talk to any of the subsequent editors of Brain and you will realize it was a 2-day-a-week commitment to manage this journal and create what it's become. It's come back to Oxford now with Masood Hussain. I don't know how John would have done it. I know that I've seen photographs of him correcting manuscripts on the beach in his holiday. But this is an immense achievement to do this on top of everything else. This is his retirement dinner, and you can see the same characteristic sort of bonhomie, if you like. That's Alan Cowie, I think. And this is the picture that Angela gave me when he retired. He had a 50th anniversary reunion. Getting wings means getting your pilot's accreditation in the RAF, and he was very delighted to be meeting up with all his old friends from those days. the department that he, I think, created, and I think that is the right way of putting it, because he was the first to really exemplify the clinical and scientific role and what could be achieved by bringing these things together. This department is now huge. It's got approaching 500 people. It's divided into 6 divisions, and there's a huge amount of activity. But the same model is there. We have a large number of people who are effectively clinical trainees, and they come through. They do research with people like Angela, David, and then they turn into people who want themselves to do both in the future. And I think that's our model, and that's how we've been successful. And I think we owe a lot to John for that. So thank you very much for listening. I'll hand you back to Marianne.
Thank you, Gavin, for putting your memories of John Newsom Davis in a historical perspective on Oxford, and pointing out that he was one amongst a number of renowned neurologists based at the University of Oxford. Thank you all 3 for sharing your beautiful memories of John Newsom Davis with us. I must say I am very grateful as a clinician to what John Newsom Davis has brought us, because in the clinic we can use his diagnostic and therapeutic armamentarium, still we do, by the antibody testing, and Kevin showed a slide on the prednisone and azathioprine trial. We still use that as a combination therapy in patients with myasthenia gravis, the plasmapheresis and IVIG. So what he has brought us is still in use in the clinic, and we have to be grateful for that. Angela, you told us about the thymectomy trial. It has been said that he had to travel to NIH time and again. I was told 10 times in total, in order to convince the people at NIH that this project was really worth a grant. So he really believed in this research question. And I met John only once, when, and that was quite a long time ago, when he was invited to give a lecture at the annual meeting of the Amsterdam Neurological Association. And not only did he deliver a splendid lecture, but also at a dinner party afterwards, we noticed that he was not only a very kind, but also a very witty man with a good sense of humor. So I have fond memories of him too. I'm sure that there will be people in the room who would like to comment on personal contacts with or other memories of John Newsom Davis. I know that Professor Singhal would like to comment. Professor Singhal?
Yeah, I've unmuted myself, and thank you very much. Very kind of you indeed. I really enjoyed the complete session and really overjoyed. And we are very, very fortunate to have Professor Newsom Davis come to India on our invitation to address in Neurology Update in the 2002. Despite a little bit of touch of laryngitis that he had, he gave a spellbound lecture, which were attended by several students from India, and they were really greatly impressed. We again invited him on behalf of the Indian Academy of Neurology in the city where Dr. Meshram lives in Nagpur, and he gave an outstanding lecture. He was very, very warm, very friendly. He put us at ease all the time, and he was very kind even to give us his slides, to use it for the students' lectures. In fact, he was very kind also to guide us in our very, very difficult cases. We would get in touch with him on telephone or write to him, and he would reply to us. I also remember one Air India patient who had limb girdle myasthenia, and we were not able to really handle him. And with Professor Beason's help, I think they could identify the gene defect and we could treat them. Oxford really has become a center of excellence for us. I must say that we have used it often from all over India. We send specimens to Oxford to get our samples tested. Yeah. Hello. And I think even Professor Angela Vincent, was very kind on our invitation to come to our meeting in Mumbai at the Thailand and also give us a book. And not only that, I'd like to thank Professor Vincent for helping Dr. Nisha when we had sent her to you and to learn techniques and how to do it. And that made a lot of difference in India. In fact, many of the laboratories, you were always wanting that somebody should come and learn the technique. We still use your laboratory at this Oxford for many of the samples of the patient with autoimmune disorders of varied type. Thank you very much indeed, and I'm really grateful and have very fond memories of my friendship. I might say friendship with Professor Non-Duwas. He was very high up and still never made me feel so low. And he was kind enough even to invite us during the World Federation of Neurology meeting in London to his house on the Thames. Once again, I visited him at his house when his wife and he were very, very warm and very, very friendly and very, very kind. I have very fond memories of Professor John Newsom Davis and miss him very much indeed. Thank you.
Thank you very much, Professor Singhal, for sharing your memories of your friendship and your collaboration with both John Newsom Davis and Angela Vincent with us. Thank you very much for that. Are there others in the room who would like to make a comment that might have collaborated with John? We have Rad Shakir, Wolfgang Grieselt.
Yes, if I may say something, Marianne. My first encounter with John Newsom Davis was at the World Congress of Neurology in Hamburg. And we were in a small meeting about paraneoplastic syndromes. And then he asked questions and I asked questions and he looked at my badge. And since that, we've been talking. And I've consulted him several times. And as the Austrian Society now has its 20th birthday of neurology, I was inviting him for the opening lecture in Vienna in 2001. And so it was a continuous flow. And I have very fond memories of his excellence and also of his kind ways to communicate. Thank you.
Well, thank you,
Professor Raj Shakir would like to say something.
Thank you. My memories of John Newsom Davis are really vivid and great. I chaired the local organizing committee of the World Congress in London in 2001. And that was the time, we're very lucky to have it because it was 3 months before 9/11. And it was supposed to be in September, but we couldn't get Earl's Court. So to our luck, the meeting came forward 3 months. So when I met John for the Congress, and we talk a lot, and he really, when I asked him about issues regarding my training and, sorry, can you hear me?
Yes, yes. Sure.
Oh, right, because there was he was generous, he was kind, and he asked me, because I trained with Ian Simpson. Those of you who know or don't know Ian Simpson, he was I think his paper in 1960 was the first paper to say that myasthenia is an autoimmune disease, was published in the Scottish Medical Journal of all journals. And when I told him where I trained in Glasgow, he reminded me of that. And that was very kind of him to talk to me at the time. This was my first time to meet him, but I met him several times. Very kind and truly an honor to talk to him at that time. And I will never forget our encounter when he congratulated me on how the World Congress local organization went in London in 2001. Thank you.
Well, thank you, Rad. It's amazing to see how many people have met John Newsom Davis have collaborated with him, have benefited from his wisdom and his advices with regard to patient care.
Professor Hadimanji.
Hi, great, thank you. Thank you for 3 wonderful tributes, and it's very nice to see some photographs. And I recognize, Angela, from your talk, Giles Eldrington in one of the photographs, and also Tassie Chang from Colombo, who's I think on this call here. I never worked for John Newsom Davis, but I did apply for a job in Oxford, which was linked into High Wycombe, which is a general hospital not far from Oxford. And I went to see him about this job. And as Kevin said, you know, he was very good for giving advice and mentoring people. He put his hand on my shoulder and said, just very simply, this job is not suitable for you, and I don't think you should apply for it. And in fact, it was absolutely the right advice for me at that time. Angela, there was amongst the registrars sort of a rumor that when John was a junior at Queen Square, he was advised by a very senior professor that neurology was not the right specialty for him. Is that correct, or is that hearsay?
As to the best of my knowledge, it's absolutely correct. But that person was not the easiest, and I have to say that actually that person was responsible for my failing my clinical finals the first time.
I actually was his houseman for 6 months at the Middlesex, and he never spoke to me for the whole of the 6 months because he never spoke to the houseman. He only spoke to the registrar. My role on the wall-brand was to carry the neurological tray, and that was my role in life.
Well, I didn't include because of time, and I took too long anyway, and I apologize for that. But there was this story about John's book, you know, the book on respiration that he co-authored, which was presented to the same person that we're talking about, which was very kind of John, considering he had been told not to be a neurologist, and then was found in a secondhand bookshop by, I think I can say, Professor Alistair Compton at a later time, clearly discarded, which John was very cross about.
Great, thank you.
But there's always somebody in neurology that one can complain about. Not many, I hasten to say.
Thanks.
Not many at all.
I'd like to thank all the speakers. With that, I would like to hand over the chair to Dr. Meshram.
Yeah, thank you. Any more comments from people in the room? Anybody would like to say something? Okay, I think we had a great session as expected. I think the series is going on very well. People are enjoying. Today there might be. Steve would like to say something.
No, sure, thank you. I just want to thank Professors Vincent, Beason, Talbot, and De Visser for just an incredible session. I did not have a chance to meet Professor Newsom Davis, but I've learned so much about him today in the history of myasthenia, Lambert-Eaton, neuromyotonia, congenital myasthenic syndromes, functional MRI. It's just been a remarkable day. And Dr. Meshram, you put together just a remarkable series, and today was just a tremendous, tremendous event. So thank you so much.
Thank you, Steve. I think we had a great session, and I would like to thank Professor Angela, Vincent, David, and Kevin, and the great, historical perspective of not only John Newsom Davis but the Oxford and we could go back to the memory lane and visualize the things in a right perspective. And I would like to thank Professor Mariam Visser for chairing the session, and Professor B.S. Singhal, Professor Griswold, Professor Raj Shakir, Steve Lewis, Professor J.M.K. Murthy, Professor Sarosh Katrak to be with us, and all the participants. I know there most of the neurologists are put on COVID duty because the India is facing shortage of doctors. So, and I think we'll overcome this. And the next session, I just want to remind that it is about Professor Raymond Adams, and Professor Ellen Roper will speak, and we'll have interaction with Raymond's daughter, Nina Salam, and others, and Professor Martin Samuels will chair. And after that, Professor B.S. Singhal will speak about Professor Nosher Wadia, and in the month of July, on 3rd July. And in August, we'll have Peter Gorsby to talk about James Lance. And then in November, it will be Perry-Marie Preux and Amado Gallo-Diop and Michel Dumas. So interesting sessions ahead. So be with us on the first Saturday of the month. Thank you very much. Thank you all participants to be with us. And thank you once again. I would like to thank all the speakers and the people in the room. Thank you very much.
Thank you.
Thank you very much.
Thank you.
Thank you. Thank you, Marianne.
Thank you.
Thank you. Thank you, David. Bye.